let's. I'm doing a fast speed one through the biology flashcards in for like 17 minutes or less, and let's do it. So, an allele is a what form of a what located at the same position on a chromosome. You can pause the video, for example. You hear me saying this, so this is how I'm gonna do it for myself. And if I know the answer, I play it to see if the answer, what is the answer basically. So again, an allele is a what form of a what located at the same position on a chromosome. It is a variant form of a gene. Just had this card earlier in the day, so this is a hard card for me. Genotype is what one has at what. It's the combination of alleles one has at a given locus. A homozygous genotype has what alleles? It has two of the same alleles. Yes. A heterozygous genotype has two of the different alleles. Two different alleles, yes. A what allele is only expressed if an organism is homozygous for that trait. And a what allele is only expressed if an organism, so it's a recessive allele, right? Phenotype is what? It's the demonstration and representation of the genotype. Observable manifestation of a genotype. Complete dominance is a form of dominance. So we have complete dominance, which is like, is it a dominant allele? In heterozygous condition, wherein the allele that is regarded as dominant completely masks the effect of the allele that is recessive. It completely masks does the dominance, right? So that would be the dominant allele completely masking the effect of the allele that is recessive. By masking, it means that dominant allele gives us a heterozygous phenotype, which is the same phenotype as the homozygous dominant phenotype. So the other two types are codominance and incomplete dominance. So codominance basically results in the B and like blue and yellow to have dots on it. It's like semi thing. And incomplete is just completely, it's like the total combination of those things, which is not blue, not yellow, but it's green. If need be, we can go more in depth into that, but let's do it good for now. Incomplete dominance is a form of what inheritance in which one allele for a specific trait is not completely expressed over it. So incomplete is the green, right? Has to have, so is not completely expressed over its paired allele. Has to have two homozygous of different alleles to let this happen. Is a form of intermediate heterozygous inheritance. You essentially make a third trait a new color, for example, whereas codominance is more like blood type. It's tricky. You need to remember that incomplete results into the intermediate heterozygous. Codominance is in which a representation of both alleles is demonstrated, represented, right? An example is the blood type. Both alleles are simultaneously expressed in the heterozygote. So we can remember that. Penetrance is the probability. What is penetrance? It's like the penetration of an allele into the population, like, so the probability of having a specific allele, the proportion of individuals carrying a particular variant of a gene, aka alleles, that also express an associated trait. Measures how often a phenotype occurs. So we also have complete penetrance and incomplete penetrance, and also it's related to expressivity. Penetrance and expressivity describe how genes are expressed as phenotype. Penetrance is the percentage of individuals having a particular genotype that express the expected phenotype. It measures how often the phenotype occurs. And expressivity is the degree to which a character is expressed. It measures the intensity of the phenotype. Both examine how gene expression is affected by environment and genetic background. We have variable penetrance, variable expressivity, variable penetrance, and expressivity. This was a bit confusing since it's only dark purple and white for variable penetrance. For variable expressivity, you no longer have white, right? You have degrees of purple, and both of them, you have white, you have degrees of purple, and you do also have the dark purple. So how often the phenotype occurs? And expressivity, the intensity of the phenotype. So it's a degree, right? To what degree you're expressing it. But penetrance is just the proportion of individuals carrying that allele that also express an associated trait. So complete penetrance with broad expressivity, you have degrees. Incomplete penetrance with limited expressivity is either white or purple. So complete penetrance is more like complete dominance, and incomplete penetrance is the degrees with broad expressivity, right? But since it's incomplete, you don't have that much of the mutant phenotype. You do have wild type too. Cool. So remember, the mutant phenotype is what is the proportion that we're talking about. Variable expressivity is when what happens. So variable expressivity, as we just saw, is when the different intensities of the gene is expressed. So different intensities of the phenotype is when the phenotype is expressed, but to a different degree among individuals with the same genotype. Measures intensity of the phenotype. Remember, intensity and expressivity. Genetic leakage is when what happens. Is it the fact that suddenly those genes that were heterozygous, they start to appear, be represented, expressed? Genes flow from one species to another via hybrid offspring. Is when genes flow from one species to another and still hybrid offspring. And that's why we have a zebra horse. genes flow from one species to another via hybrid offspring. And what is genetic drift? It's when the composition of the gene pool changes as a result of what? It's as a result of chance. So, what is the difference between genetic leakage and genetic drift? Genetic leakage and genetic drift are distinct concepts. Genetic leakage, as we just saw with the zebra horse, is gene flow. Occurs when genetic material, genes, is exchanged between different populations of the same species. This can happen through the movement of individuals or gametes, reproductive cells between populations. Migration or interbreeding between individuals from different populations can lead to transfer of genes. This process tends to homogenize the genetic makeup of populations over time, making them more similar. Genetic drift is a random process that can lead to changes in the frequency of certain alleles in a population over time. It is driven by chance events rather than natural selection. Ooh, so genetic leakage is kind of like natural selection, the interbreeding that happens. But genetic drift is totally by chance. And it is more pronounced in smaller populations. Random events such as death or reproduction of a few individuals can lead to a shift in allele frequencies. Since these events are random, the changes in genes frequencies are not necessarily adaptive. Cool. The founder effect. What is the founder effect? It's the loss of genetic variation that occurs when a new population is established by a very small number of individuals. So you have a very small number of individuals and a new population is established by them, and then you have the loss of genetic variation. So that's when the bird is just taking the genes into a new field, and all you get is the alleles for yellow flower color. This is the founder effect. Inbreeding, as they say. So interbreeding was genetic leakage, the zebra horse, and inbreeding is the founder effect. Mendel's law of independent assortment states that so this is related to when the homologous chromosomes are exchanging their genes. So independent assortment is that like the chances of the assortment of the alleles of two or more different genes get sorted into gametes independently of one another. So yeah, the odds of the genes being sorted into gametes is independent of another, right? In other words, the allele a gamete receives for one gene does not influence the allele received for another gene. That's the independent assortment law. Now, law of segregation is that each hereditary characteristic, so allele, is controlled by two alleles which separate into different gametes. Yes. And segregation, the gametes are segregated. Two of them determine the hereditary characteristic that you have. And again, independent assortment is the fact that the chance of one allele being in your body is not affected by the other allele from the other gamete, right? So it's independent to allele separation for another gene. And the principle of dominance, we had complete dominance, stuff like that, codominance, incomplete dominance. In pairs of alleles that are different, one allele will mask the effect of the other allele. These laws are known to be due to key events that occur during meiotic division, yes, for our gametes. Okay, let me just finish this right here. The law of segregation describes how homologous chromosomes and hence allele pairs are separated in meiosis 1. The law of independent assortment describes how homologous pairs align randomly as bivalents during metaphase 1. So it's in where the plates meet. It's in the middle, right? When the homologous chromosomes are in the middle. And they're independently assorted. Through the elucidation of the process of meiosis, we know that there are certain exceptions to Mendel's laws. Genes that are on the same chromosome, linked genes, will not undergo independent assortment. So the exception is actually this: the linked genes that are on the same chromosome will not undergo independent assortment unless recombination occurs. So they are actually the chances of the linked genes, the chances of them being expressed. Is it about expression, or is this about just transferring to the next generation? It's more like the meiosis that we're talking about, right? And so linked genes will not undergo independent assortment unless recombination occurs. Not all genes display a dominance hierarchy. Certain traits may display codominance or incomplete dominance. Yes. When you discover the fundamental law of genetics by being a pea garden farmer, it ain't much, but it's honest work. Law of independent assortment: alleles for one trait segregated independently from alleles for another trait in metaphase 1 of meiosis. Each allele combination is equally likely to occur. Exception to this rule is linked genes. So law of independent assortment is that when the two alleles are separate from independently get assorted from another, they do not affect each other's chance of being sorted into gametes. Okay, I'll be right back.